Hi all, we are looking at question number 23 from GATE Biotechnology 2017 paper. So the question is, the transcription factor X binds a 10 base pair DNA stretch. So in the DNA of an organism, X was found to bind at 20 distinct sites. An analysis of these 20 binding sites showed the following distribution. So they've given a table for each base like A, T, G and C, and a score for each of these bases across 10 different positions in the binding site. So what is the consensus sequence for the binding site of X? So the options are 4 different nucleotide sequences, and N here means that at that particular position it could be any of the following or any of these 4 bases. So before we answer the question, let's briefly learn about transcription factors and what sequencing technology is used for identifying these transcription factor binding sites, and about the table that's given in the question. So in molecular biology, a transcription factor is a protein that controls the rate of transcription of genetic information from DNA to RNA by binding to a specific DNA sequence. So briefly, the transcription of an mRNA from DNA is controlled by a lot of transcription factors and cofactors by their binding to the promoter site of the DNA sequence. So the DNA inside the cells are condensed and are packed with the help of proteins called histones. So this packaging allows the 3 billion base pairs in each cell to fit into a space of just 6 microns. So if you stretch the DNA in one cell all the way out, it would be about 2 meters long, and all the DNA in all our cells put together would be about twice the diameter of the solar system. So chromatin, which is the complex of DNA and these histone proteins, undergoes dynamic changes that influence the accessibility of these genes. So the part of the gene, the segment of the DNA that's bound with the histone is not accessible for transcription or translation, whereas these free segments, they are free to be bound by other factors and hence to initiate transcription and translation. So histones play a central role in organizing DNA into nucleosomes and higher order chromatin structures. So transcription factors bind to specific DNA sequences called transcription factor binding sites in the regulatory regions of the genes. So by regulatory regions, it mostly means the promoter regions, which is like upstream to the transcription start site. So these binding events serve as key regulatory switches that control whether a gene is activated or repressed. Transcription factors can influence transcription directly or indirectly. For example, transcription factors play a critical role in recruiting RNA polymerase, which is the enzyme responsible for synthesizing RNA from a DNA template at the transcription start site of the target gene. So this recruitment is facilitated by interactions between the transcription factors and several other cofactors and the general transcription machinery. So the transcription factor can attract such cofactors and enzymes, thereby promoting transcription, or can block the RNA polymerase from initiating transcription, thus repressing the genes. So this is important in different cell types. For example, transcription factors promote expression of genes relevant to the brain cell functions in the brain, while these genes are repressed in other tissues like in the heart. So the cells in the heart do not need to perform the functions that the brain performs. So yeah, this gives cell type specificity for genes and the tissues. They can also act indirectly by influencing chromatin remodeling. So pioneer transcription factors are a type of transcription factors that have a unique ability to bind to their target DNA sequences even when these sequences are embedded within nucleosomes, like you can see here. So the binding of these pioneer transcription factors to the nucleosomal DNA can lead to the displacement or partial unwrapping of nucleosomes. So this initial step is essential for creating a more open chromatin structure that allows access to regulatory elements. And transcription factors can also bind the accessible DNA and prevent them from recondensing into nucleosomal structures. So this is how transcription factors interact with the DNA and the nucleosome complexes, and how they influence transcription. Now, how do you identify the sequences or the motifs where the transcription factors are bound? So, for these, there are specific sequencing methods for this purpose, like ChIP-seq or ATAC-Seq, and so on. So, let's get an idea of how ChIP-seq works. So, ChIP-seq stands for chromatin immunoprecipitation. So, ChIP-seq is a widely used method for identifying genomic regions where specific proteins like transcription factors interact with the DNA. So first, the sample is cross-linked with the transcription factor of interest. So the cells are treated with a cross-linking agent, usually formaldehyde, which covalently links the transcription factor to the DNA. So this step freezes the protein-DNA interactions in place, preserving the physical associations between the transcription factors, histones, and the DNA. And then the DNA is fragmented. So now we have the transcription factor bound to the DNA. Now, they are extracted using antibodies that are specific to the transcription factor of interest. So, the cross linking is then reversed and the transcription factor is separated from the DNA. So, these free DNA fragments are now sequenced and mapped to a reference genome, or they can be hybridized using a microarray. So from sequencing these DNA, we get the composition of the fragments. So in our question, the transcription factor had 20 binding sites, and hence we get 20 sequences, each 10 base pairs in length, composed of A, T, G, and C. So a multiple sequence alignment is done and something called a position weight matrix is generated, and that was the table that was given in our question. So it basically tells us how many times a particular nucleotide is found at a given position using all these binding sites. So for example, here you can see ATGC and positions 1 to 10 and values ranging from 0 to 20 because we have a total of 20 sequences which are the transcription factor binding sites. Now let's read this table. So 11 means at position 1, 11 sequences out of these 20 have A, and 3 sequences have T at position 1, 4 sequences have G at position 1, and 2 sequences have C at 1. And looking at position 2, so all the 20 sequences have G at position 2. So that's how you read this table for each position. The base with the maximum number is the actual base that is found in the binding site of the transcription factor. Now coming back to our question, so we need to analyze the 20. So they've given the position weight matrix for the 20 binding site sequences, and we just have to identify the sequence of the binding site. So the base at the first position would be A because maximum sequences have A at position 1, and the second position would have a G, and third position has T, and fourth position C, position 5 A, position 6 C, and position 7 it has equal numbers for G and C, so it could be either G or C, and so we represent it by N. And at position 8 all the 20 sequences have T, and at 9 G, and the last base is C. So deducting the sequence, option B would be the answer. So we would have A, G, T, C, A, C, N, T, G, C. So that answers our question. So this is the sequence for the binding site of the transcription factor X.